and welcome back to the Sound of Science. Today we're going to be talking about the process of meiosis. Meiosis is just simply the creation of gametes or sex cells like sperm and egg. And it's different from mitosis because it really needs a different outcome. It's trying to achieve a different outcome. So I'm going to start by reminding you what we learned about mitosis, and then we're going to go into meiosis. So in mitosis, we talked about how the cell, all your cells in your body, your somatic cells, have these two complete copies of your chromosomes. They come from mom and dad, right? Mom gives a copy of chromosomes to you, and dad gives a copy of your chromosomes to you. And so you have two sets of chromosomes, one from each parent. And during the course of mitosis, we're going to make a copy of each of those chromatids. We are going to copy everything in your cells, and then it's going to divide in such a way that every cell that's created is identical. So mitosis is creating identical cells, and in fact is going to create two identical cells. In addition to that, it's going to go through one division, and it's going to end up with a complete set of chromosomes numbering 46. So that's information that you really need to know about mitosis. So now let's compare it to meiosis. And by the way, you should know the differences between the two. Maybe even take the time to write those down as we go through. So for meiosis, we're actually going to go through two complete cell divisions rather than just the one. And we're also going to end up with 4 non-identical cells. So the 4 cells that are being produced at the end of all of meiosis are actually going to be completely genetically different from each other. In the end, they also are only going to have one set of chromosomes, so we're going to refer to them as haploid, whereas in mitosis, because it has 2 sets, we refer to that as diploid. So make sure you sort of understand that. And of course, like I said, in humans, 46 is the number of chromosomes we have in our diploid cells. Our haploid cells, our sex cells, are going to have only 23. Okay, so to help you sort of see some of this, what's the difference between haploid and diploid? Well, haploid cells, like I said, are going to have non-homologous chromosomes and only one set of it. So you end up with 23 chromatids or chromosomes that code for different things. Remember, this is DNA. Chromosomes are just DNA, guys, and it codes for proteins. It makes our traits. We're going to learn a lot more about that in the coming days. So if you have a diploid cell, it's going to have 2 copies. So for instance, let's take a look at this pink chromosome. So in the haploid cell, like that pink chromosome is going to have the genes for your hair color and hair structure, right? In the diploid cell, the pink chromosomes are still going to have the genes for your hair structure and your hair color, but it may be 2 different types. Like for instance, one of them might say to have curly hair that's blonde, and another might just say have straight hair that's brown. So it really just sort of depends on the genes that you get from your parents. And those genes are going to actually work together in order to give you the traits that you have. Again, I'm getting ahead of myself. This is all genetics, but I just wanted you to kind of understand. Even though those 2 pink chromosomes are coding for perhaps a different version of that gene, they both have the same genes on them, and that's how your chromosomes work as well. Okay, I'm going to very quickly go through meiosis 1. Now, this is the information that's going to hopefully help you understand the different way that it works. But if you find yourself confused at the end of this, don't worry. I am actually going to go through sort of a pictograph of it as well that might help. So just like in mitosis, meiosis actually has prophase, metaphase, anaphase, telophase, and cytokinesis. The difference is we're going to do it twice. So instead of just saying prophase, we're actually going to say prophase 1. Our chromosomes are going to make copies. They are going to match up with other chromosomes that are similar. And actually, they're going to form something called a tetrad. So in mitosis, if you think back, the two chromosome pairs or the two chromosomes sort of got side by side, right? And they lined up during metaphase. So here in meiosis, initially, you're actually going to have two pairs that are going to line up together. And so this is what we call a tetrad. It's called tetrad because tetra means four, and there's four little chromosomes, right? So they're all going to line up together. And during metaphase, they're actually going to line up in the middle of the cell. So you might have several tetrads. Then during anaphase, they're going to split apart and move away, just like this, right? So you're going to end up with what looks like normal chromosomes pairs on either end of the cell. During telophase 1, they're going to have the nuclear membranes form around them, and we're going to go through cytokinesis. So now we have two cells that look pretty normal. They look diploid. They've got two copies of chromatids and all of that fun stuff. During this phase where they're all lined up like this, sometimes they can actually cross over. When they cross over, sometimes they swap DNA. So you actually end up with DNA from one chromosome on another chromosome. This gives us infinite possibilities when it comes to genetic variation. We're going to find out a little bit later how important that is. So during meiosis 2, remember we're sitting here, we've got these two cells hanging out doing their thing. They're going to do basically the same thing that we see in mitosis. So and I'm just going to move my picture back down. During prophase 2, the chromosomes again are going to become visible. And they went back into that hairball chromatin form. During metaphase 2, the chromosomes are going to line up in the center just like they normally do in mitosis. Anaphase 2, the chromatids separate and move away. Telophase cytokinesis, we're actually going to separate all the way. And because this is happening in both of the cells we created in meiosis 1, at the end we get 4 cells that are different, which is what we're looking for. So for all of you who kind of followed this and now you're just a little bit confused, let's look at the picture. It might make a little bit more sense. So if you look here, this is just showing you two examples of the same thing. We've got gamete formation in males and gamete formation in females. So we're going to start off with the cell just being a cell, living its best little cell life, and then it's going to start going through meiosis. And it's only special cells that do this. So do not worry, you're not going to be creating gametes from some random cell somewhere. These are cells that are specifically designed to do this. So prophase is going to occur. The nuclear membrane breaks apart. We start to get our chromatids all stuck together in their little tetrads. We're going to have crossing over occur, which increases genetic variation. Hey, make sure you know that. Make sure you write that down. Crossing over occurs in prophase 1, and it's going to increase your genetic variation. We're going to go through anaphase 1, where they're going to separate and they're going to move. And here you can see, if you take a look at this picture, you can actually see how it looks like two little, chromosomes together going off to either side. It's going to go in through cytokinesis, telophase and cytokinesis, and then it's going to turn back into a hairball format and immediately go into meiosis 2, which is going to give us that look that looks a lot more like mitosis, actually. But at the end, we're going to have 4 copies that are all different. So we're going to have 4 cells, and they're all different from each other. Whereas again, in mitosis, it's 2 cells, and they're both the same. So you can see how these phases work. Now, am I going to ask you on a test to identify, is this cell in meiosis 2 anaphase, or is it, anaphase from mitosis? No, I'm not going to do that. That's honestly a lot more challenging to identify than you might think. So we're not going to end up doing anything like that. Instead, what's going to end up happening is you just really need to know the differences between the two processes, like I said, and you just need to understand the general idea of what's happening, as well as where crossing over is occur. So as long as you know those things, you're good to go. So make sure you go back. You have those things written down in your notes. In the next couple of days, we're going to be talking a lot about genetics, and we're going to talk about how this process actually leads to increased genetic diversity for all of the sexually reproducing organisms. So until next time, thanks for watching.